Hello guys, I am Shubham from Tutorial. This video is all about the nucleic acids. So before we start, subscribe to our channel and press the bell icon to get notified whenever new videos out. This is absolutely free from your side, but this will help us grow. Nucleic acids, basically we know that DNA and RNA. These are the nucleic acids. Deoxyribonucleic acid and ribonucleic acid, which have the genetic information. Okay, genetic information is stored in this molecule, right, of a organism. It may be plants or animals, bacteria, viruses, right? Nucleic acids are made up of nucleotides. Okay, nucleotides. So first of all, we have to know about nucleotides, and then we'll go to how nucleic acids are formed. So nucleotides are the building blocks of nucleic acid. Nucleotides क्या है basically? This is consists of three components. First of all, ribose sugar. Okay, ribose sugar plus nitrogenous base and phosphate group. Phosphate group. Okay. Now this ribose sugar is basically deoxyribose in the case of DNA and only ribose in the case of RNA. Right. Nitrogenous base basically nitrogen containing molecule. Right. Nitrogen containing. Okay. There is one more term known as nucleosides. Okay, nucleosides. Basically, this is nucleotides. In simple words, nucleotides minus phosphate group. Okay, got it? न्यूक्लियोटाइड माइनस फॉस्फेट ग्रुप मतलब न्यूक्लियोसाइड्स में सिर्फ राइबोस शुगर एंड नाइट्रोजन बेस प्रेजेंट होते हैं. ओके, phosphate group is absent. When phosphate group is attached, it becomes a nucleotide. Got it? Now let's understand the biochemistry of this molecule with the help of pictures. It will be more easy. So this is a ribonucleotide, basically present in RNA, and deoxyribonucleotide present in DNA. Okay, chemical structure very important. See, 2' OH is present in ribonucleotide. Okay, and when oxygen is removed, that is deoxy form. Okay, deoxy form you can see here. Right, on the second carbon of ribose sugar. So this is a sugar. The ring structure which you can see, this is a sugar, ribose sugar. Okay, and this is base. Base is basically nitrogen containing. Right, we will see it. And this is a phosphate group. Right, now the bonds which are important. See, this is nitrogen containing base and this is ribose. इसके बीच जो bond होता है, that is N glycosidic bond. Okay, I will write here N glycosidic bond. Okay, same. And जो phosphate group एंड शुगर के बीच में जो बॉन्ड है, दिस इज फॉस्फो डाईईस्टर बॉन्ड. फॉस्फो डाईईस्टर बॉन्ड. Same for here as well. Okay, do note अभी हमने मॉलिक्यूलर बायोलॉजी में रेप्लिकेशन ट्रांसक्रिप्शन देखा है, राइट? तो डी प्राइम ओ एच हमें पता है, दिस इज कॉमन एंड विच इज ग्रोइंग एंड, राइट? विच इज ग्रोइंग एंड 5 प्राइम टू 3 प्राइम डायरेक्शन. ओके, तो 5 प्राइम इज फॉस्फेट, राइट? 5th कार्बन पर फॉस्फेट ग्रुप अटैच होता है एंड 3 कार्बन पर ओ एच ग्रुप एंड यहाँ पे एडिशन होता है. So this is common for both ribo as well as deoxyribonucleotide. Whereas difference is in the 2 prime position, that is second carbon position. So this is easy to remember. Now talking about nitrogenous base. Okay, nitrogenous base compounds, 2 types, purine and pyrimidine. Right? Purine छोटा नाम बड़ा रिंग स्ट्रक्चर एंड pyrimidine थोड़ा सा बड़ा नाम सिंगल रिंग स्ट्रक्चर. Okay, like this you can remember. So purine is 9 molecules are there, whereas in pyrimidine 6 membered ring present. Okay, 6, whereas in purine it is 9. Right? Now in purines, adenine guanine, purines, and in pyrimidines, cytosine, amine, uracil. Okay, amine is present only in DNA, uracil is present only in RNA. Okay, now how to remember this purines and pyrimidines? Purines, Ag, and pyrimidines, cut. Right? Purines, Ag, adenine and guanine, pyrimidines, cut. Cytosine, uracil, amine. Now from this picture you have to remember one thing. Guanine was first isolated from guano, that is bird manure, and thymine was first isolated from thymus tissue. Okay, sometimes this small information is asked in the exams. Okay, directly questions उठाते हैं. Now this is the table you can see of nucleotide and nucleoside, their presence in the nucleic acid. So you can see जो purines है adenine, guanine, deoxy forms. Okay, deoxy, adenosine and deoxyguanosine DNA में present होगा. Adenosine and guanosine RNA में present होगा, nucleoside form. Okay, nucleoside का naming है adenosine, SINE comes, and nucleotide adenylate or guanylate, or you can say adenylic acid. Okay, guanylic acid and deoxy forms respectively. Right? Same we are with pyrimidines. Okay, cytosine, amine and uracil. Now amine present only in DNA, so deoxy form will be more prevalent, right? Deoxy form ज़्यादा present होगा amine का, so it is amidine or deoxythymidine, nucleoside, amidylate or deoxythymidylate, nucleotide form. Uracil, which is present only in RNA, you can see only uridine, uridylate. So this is the naming. Okay, nomenclature. See, it is important. So you can check the table and abbreviations, the structures. Again, this is a same showing the comparison between deoxyribonucleotide and ribonucleotides. Okay. About this structures, we know that DNA is more strong. Okay, DNA is more strong, basically stable than RNA. Okay, why? Because of this thymine group present in DNA only. Methyl group. Methyl group is present only in thymine, which is present in the DNA only. So it gives stability for DNA, right? As well as deoxyform. First is methyl group, second is deoxyform. Okay, तो राइबोस शुगर है deoxyform है. Deoxyform on the second carbon. So this is the next reason for this stability. Okay, in the coming section you will understand. Okay, now this is conformation of ribose, right? Aldehyde sugar है, ring structure beta furanose, right? Beta furanose. So this is C2 endo and C2XO. Okay, endo मतलब ऐसा ऊपर साइड है, right? Facing upwards. You have to remember this. Important है. C2XO and C2 endo. Next picture you can see C3 endo. Okay, facing this side C. And नीचे जो है that is C3XO. Got it? Endo is basically facing upwards. XO is downwards. It's shadow like. Some minor purine and pyrimidine bases shown as nucleosides. 5-methylcitidine occurs in the DNA of animals and higher plants. And 6-methyladenosine in bacterial DNA. 5-hydroxymethylcitidine in DNA of animals and of bacteria infected with certain bacteriophages. Okay, direct information. Some minor bases of tRNAs. Right, tRNA is present as pseudoutidine. Okay, inosine contain the base hypoxanthine. Right, note that pseudoutidine, like uridine, contain uracil. They are distinct in the point of attachment to the ribose. Okay, point of attachment is distinct. In uridine, uracil is attached to N1. Okay, you have to note these differences, right? uracil में N1 नाइट्रोजन 1 पर अटैच होता है. राइबोस the usual attachment points for pyrimidine. In pseudouridine, uracil is attached through E5. So this is the important information you have to highlight. Okay, uracil attached through N1 in uridine, right? And pseudouridine में uracil is attached through C5, fifth carbon. These are some adenosine monophosphate forms, right? Adenosine 2' monophosphate. 2' means on the second carbon phosphate group is present. Adenosine 5' monophosphate means fifth carbon पर phosphate group present है. Adenosine 3' monophosphate means 3 carbon पर present है. And adenosine 2', 3' cyclic monophosphate means both of these are involved. 2 and third carbon. Right. Now this is how DNA and RNA form nucleic acid basically from nucleotides. Okay. You can see one end is 5' end and other is the 3' end. Okay. DNA contains of thymine, RNA contains of uracil. Other things are same. Okay. So how is the bond formation? 5' to 3' position. Right. It is third carbon पर ऐसा linkage होता है. This is basically phosphodiester linkage. Common in both DNA as well as RNA. right? Now this is the next picture where you can see hydrolysis of RNA under alkaline condition. Okay, 2' OH present only in the RNA ribonucleotides is prone to attack, right? Prone to attack and reactions take place. Okay, see here. The 2' hydroxyl act as a nucleophile in an intramolecular displacement. The 2' 3' cyclic monophosphate derivatives is further hydrolyzed to a mixture of 2' 3' monophosphates. DNA which lacks 2' hydroxyl is stable. This is why DNA is stable, because it is not having OH group, it is having the deoxy form. Okay, so there is no nucleophilic attack seen in the case of DNA, as well as methyl group present in the amine and DNA gives more stability, additional stability. Just a small information, uracil tautomeric forms lacteme, lactam, double lacteme. The lactam form predominates, okay, at pH 7. Other form become more dominant as pH decreases, right? pH 7 पर lactam form predominate करता है. Okay, other free pyrimidines and free purines also have tautomeric forms, but they are more rarely entered. So lactam form of uracil is more predominating. Watson and Crick, we know that DNA double helix structure was given by Watson and Crick. Okay, we know. So Watson and Crick base pairing भी है, 3 pairs with C, 3 hydrogen bonds, and A pairs with C with 2 hydrogen bonds. Okay, so double helix structure is how one strand is from 5' to 3', other strand is opposite 3' to 5'. Got it? So this is anti-parallel nature, right? So positions of hydrogen bonds formed, you have to note 6, 4, 1, 3, 2, 2. Now all these three are present in GC, but 6, 4 and 1, 3 present in AT. Okay, you can see here A and T, 6, 4, 1, 3, right? And in the case of GC, 6, 4, 1, 3, 2, 2. So before Watson and Crick, Rosalind Franklin used X-ray diffraction pattern and showing that double helix is the structure of DNA. Okay, this is also very important information. So the spots forming a cross in the center denote a helical structure. The heavy bands at the left and right are arise from the recurring bases. Got it? X-ray diffraction pattern, or basically X-ray crystallography studies, and say, which confirm basically, which is a proof actually for double helical structure given by Rosalind Franklin. This is a Watson-Crick model for the structure of DNA, right? Information. The original model proposed by Watson and Crick had 10 base pairs or 34 angstrom per turn of the helix. Subsequent measurement revealed 10.5 base pair or 36 angstrom, that is 3.6 nm per turn. Schematic representation showing dimension of the helix, thick representation showing the backbone and stacking of bases, space filling model. Okay, so C is the space filling model, B is the schematic representation, and this is helical. A is helical, B is peak representation, sorry. Okay, A is helical model, B is peak representation, and C is space filling model. Same things, you can see complementary nature and anti-parallel strands, right, as I told you. DNA replication, we saw semi-conservative nature, parental strand gets separated and new strand formation takes place. Okay, we know this is a semi-conservative nature of DNA replication. This is also given by Watson and Crick. So this is structural variation in DNA. Seen form, anti-form. Seen is towards the same direction. Actually, same direction में होना, like this. And this is opposite, anti. Anti is opposite. So stability is seen mostly in case of anti. Okay, anti form more stable. Okay, here we will see the instability. Basically, repulsion होगा, right? Now, three forms of DNA: A form, B form, Z form. Okay, B form is commonly for DNA. Okay, DNA. A form, RNA DNA hybrid. Okay, and double strand RNA भी में होता है in the A form, right? Now in A form you can see right-handed structure, as well as in B form you can see the right-handed structure. Only Z form left-handed structure, right? See the differences, base pairs. Then there are slight variations, diameter. Okay, base pairs for helical turn, helix rise for base pair. B form C2 endo, A form C3 endo. Okay, and in Z form C2 endo for pyrimidine, 3 for purine. And anti conformation, B and A form, anti for pyrimidine, seen for purines. Got it? We will discuss more on this after some time. Comparison of A, B and Z forms of DNA. Each structure shown here has 36 base pairs. The riboses and bases are shown in yellow. The phosphodiester backbone is represented as a blue rope. Blue is the color used to represent DNA strand in later chapters. The table summarizes some properties of the DNA forms of three forms of DNA. Now here is a structure, basically how we see palindromes and minor repeats. Palindrome is mirror images. Okay, same. See, TTAG, CAC, same from here also. Okay, direction यहाँ से same है and palindrome. Minor repeat, this is on the same side, not on the opposite side. Okay, here it is. Palindromes are sequences of double-stranded nucleic acids with two-fold symmetry. To superimpose one repeat shaded sequences on the other. And in minor repeat, it is symmetric sequence within each strand. Now this is hairpin and cruciform structure. मतलब hairpin की जब पेयर करेंगे in the opposite complementary base pairing, the cruciform structure forms. Right, hairpin when paired with each other forms a cruciform structure. You can see in the picture. This can be seen in DNA or RNA, anything. So this is Hook-Steen pairing. hook chain pairing, which is for the more than two helical structure of DNA, right? Between the three strands, between the four strands. Okay, RNA structures, single stranded RNA, you can see right-handed form. Okay, and secondary structure of RNA, there is pairing between each other, right? Single strand RNA की pair करता है complementary regions. See, single strand you can see bulge formation, internal loop formation, hairpin formation. tRNA हमें पता है the structure clover leaf model, right? This is the secondary structure and hairpin double helix. So because of this, so secondary structures है RNA. So this we can call double strand RNA structure, right? This is double strand RNA structure. Okay, and does not follow Chargaff rule. Chargaff rule. So what is that basically? So what is Chargaff rule? A plus T is equal to G plus C, right? A plus T plus G plus C is equal to 100%. Purines is equal to pyrimidine. So this is Chargaff rule, which follows for double strand DNA. Okay, and here it will be single strand RNA. It will be single strand RNA. Now let's see comparison. On comparison, B form, A form, Z form. B form is for DNA, A form RNA, and RNA DNA hybrid, right? And Z form DNA. Wide major groove, narrow minor groove. Wide minor groove, narrow major groove. Okay, major groove, not really groove. Okay, and narrow minor. Then this is right-handed. We saw right-handed, right-handed, and left-handed. B form favored at high water concentration, right? A form low water concentration, and Z form high salt. Okay, about sugar puckering. तो हमने conformation देखे ना, this conformations. This is sugar puckering. So about that, in B form it is C2 endo. Okay, now RNA में 2' OH है, so C2 endo cannot form. Okay, because of 2' OH. Here it is C3 endo. Here it varies. I guess this is mentioned in that table also. You can see here. This is important. This one. I will highlight right-handed, left-handed. This endo means and this anti. from this table. Okay, important thing. Now next thing, double strand DNA denaturation, partially denatured means half it is separated, half still bound. Okay, so partially denatured, and this is complete denaturation, separation of both the strands. So what we can see, there is a effect known as hyperchromic effect. Hyperchromic hue. Okay, as there is denaturation, जैसे denaturation होता है, as the double strand DNA gets converted to single strands, the absorbance gets increases. Okay, hyperchromic effect, single strand as double strand DNA gets converted to single strand DNA, there is increase in absorbance. Increase in absorbance at 260 nanometers. So this is hyperchromic effect. Okay. Double strand DNA light absorption नहीं करता. Okay. Single strand DNA light absorption करता है. So double strand DNA shows hypochromic effect and single strand DNA shows hyperchromic effect. Right. Now here are the graphs basically of two DNA structures. You can see Tm जो melting temperature है that is different. तो इसका कम है, इसका ज़्यादा है. Right. Why so? Basically, if a DNA contains more number of GC, right? GC pairing अगर ज़्यादा है DNA में, that will be hard to break, right? तो ऐसे DNA को टेम्परेचर ज़्यादा लगेगा. Okay, increased melting temperature will be needed. So GC pairing is more strong. G bonds are present, right? You can see from this graph, as GC content is increasing, melting temperature is increasing. This is just a UV absorption spectra of nucleic acid bases. Okay, you can see denatured DNA and this is the new form. And here is a partially denatured DNA. You can see bubble formation, right? You know this, we have seen in replication and transcription. As the DNA gets opened, the bubble formation takes place. In the case of replication, it is known as replication bubble. In the case of transcription, it is known as transcription bubble. Now, some well-characterized reactions, basically, you have to know. Okay, deamination. Cytosine deamination gives rise to uracil. Okay, deamination is removal of amino group. Okay, cytosine deamination gives rise to uracil. 5-methyl cytosine is converted to thymine. How to remember this? Methyl group is present only in thymine. तो 5-methyl cytosine है, तो इसका डीअमाइनेशन होने के बाद थायमीन में बदल जाए, राइट? तो थायमीन, methyl group is present only in thymine. Adenine deamination, hypoxanthine. Guanine deamination, xanthine. Depurination is basically जो रिंग है, प्यूरिन रिंग स्ट्रक्चर है, that is getting separated. Apurinic residue. This we have seen in a repair pathway, AP site formation. Apurinic or apyrimidinic site formation takes place when the purine or pyrimidine, that is nitrogen content base, is removed out. This also we have seen formation of pyrimidine dimers induced by UV light. Okay, thymidine dimers are seen where the cyclobutene ring formation takes place. Okay, cyclobutene pyrimidine dimers. So we know TT timers. We have seen in the repair pathway. Right? Chemical agent that can cause DNA damage. So nitrous acid precursors can induce DNA damage. Alkylating agents also. See, precursors of nitrous acid promote deamination reaction. Okay, important. Alkylating agents most generate modified nucleotides non-enzymatically, right, without using any enzyme. Alkylating agents alter DNA and nitrous acid by deamination. This is also one type of damage where you can see guanine tautomers when react with dimethyl sulfate forms methylguanine. Okay, you can see nucleoside phosphates here. General structure of nucleoside 5' mono triphosphates and their standard abbreviations. In the deoxyribonucleotide phosphates, the pentose is deoxy. Yes, we know this. Okay, we know this forms AMP, GMP, CTP, CMP, UMP, right? Triphosphate forms. You can check accordingly. In the cases of DNA and RNA, deoxyform and oxyform. Now this is the ATP molecule we know, which is having a high energy molecule, basically ATP. So most of our body dependent on ATP for energy, right? So you can see this is the ATP, adenosine triphosphate, and between the phosphates there is an anhydride bond. Okay, anhydride bond. This is the ester bond, and this will be N-glycosidic bond, right? Phosphate ester and phosphoanhydride bonds of ATP. If I show you triphosphate, this is alpha, this is beta and gamma phosphate. These two bonds are phosphoanhydride bond. This is a phosphoester bond. Okay, ester bond. And between adenine and ribose sugar, there is a N-glycosidic bond. Got it? Some more molecules, basically some coenzymes containing adenosine. So these are the coenzymes, right, which are important for regulatory functions, right? So coenzyme A, C, contains ADP, right? Then nicotinamide adenine dinucleotide. Flavine adenine dinucleotide. Okay, NAD, FAD. Three regulatory nucleotides. Cyclic AMP. See the positions. Adenosine 3' 5' cyclic monophosphate. Same with guanosine 3' 5' cyclic monophosphate. This is guanosine tetraphosphate 5' and 3'. Same. Only diphosphate is present. See, two phosphates. Okay. Coenzyme A functions in acyl group transfer reaction. And this is important. Acyl group such as acetyl or acetoacetyl group is attached to CoA through a thioester linkage. Right? NAD plus functions in hydride transfer. Okay, we have seen NADH forms. Okay, NADH forms. So that is hydride transfer reactions involvement of NAD. FAD, the active form of vitamin B2, riboflavin, functions in electron transfer. Okay. Another coenzyme adenosine, incorporating adenosine is 5' deoxy adenosyl cobalamin, the active form of vitamin B12, which participates in intramolecular group transfer between adjacent carbon. Okay, so these are the things which are related to nucleotides and nucleic acids as a whole. Okay, so that's it for this video. I hope this video helps you. If you find this video helpful, do let us know in the comment section. Like this video, share this video with your friends, subscribe to the channel and stay tuned for the upcoming videos. Thank you.